Hello, everyone. Thank you for listening in on the first of multiple in Aldevron's virtual Breakthrough Speaker Series. And I am glad to be moderating today's event, where we will hear from Dr. Laurence Cooper. A bit about Dr. Cooper. Laurence J.N. Cooper, MD, PhD, was a tenured professor at the University of Texas MD Anderson Cancer Center, and until recently, CEO of Xiopharm Oncology. He graduated from college in 1986 and received his MD and PhD from Case Western Reserve University. He is board certified in pediatric hematology and oncology and ran the bone marrow transplantation program at MD Anderson for about a decade. Dr. Cooper has received numerous awards and honors. He has also authored more than 140 journal articles, book chapters and abstracts, and is a member of many societies and organizations. He leads scientific and commercial efforts based on the genetic modification of the immune system to target cancer. Dr. Cooper, welcome, and thank you for sharing your presentation on behalf of Xiopharm Oncology, DNA plasmids for the genetic engineering of clinical grade T cells. So I'm going to have you take it away. Thank you.
Thanks, Michelle. Thanks, John, and thank you also to the Aldevron team here. I'm just delighted to be with you to speak on behalf of Xiopharm and to also really congratulate Aldevron on just an amazing growth story. Both companies work on a really important problem, and that is, how do you treat patients with cancer? In particular, how do you treat patients with solid tumors? And the solution that both companies embrace is through the engineering of T cells. And that's the story that we'll tell over about the next 30 minutes using about 26 slides. The first of those slides is a forward-looking statement because I am speaking on behalf of a public company, Xiopharm Oncology. Okay, so let's get into it on slide 3 here. So the genetic engineering of T cells, it generally speaking, falls into 2 camps that you can see on your screen. On the left-hand side is the engineering of T cells to express what's called a chimeric antigen receptor, and this is probably well known now to the majority of the audience. And one can think about a chimeric antigen receptor as a molecule that directly engages with the T cell antigen. Here's the antigen, and here's the chimeric antigen receptor. It's a handshake, if you would, or a death grip between the engineered T cell and the tumor cell. And this has actually resulted in commercial products. The poster child is against CD19 malignancies. And it has basically proven out the principle that infusion of genetically modified T cells can recycle their effector function and kill again, and take care of kilograms of tumor cells. So there's no doubt that the CAR works. The issue, of course, is how many of these antigens are expressed, because the CAR is able to look only at the antigens on the outside or the cell surface of the tumor cell. It has no ability, really, for the most part, to peer inside of the cell. But that's where most of the antigens are. So there are now engineering principles that are being applied to use T cells to express what's called the T cell receptor. Now, of course, T cells are born with a T cell receptor, but in this particular paradigm or this particular approach, T cells are engineered to express a T cell receptor, and we'll get into why in a few minutes. But one of the facets of a T cell receptor is that it can peer, if you would, inside of a tumor cell, because it interrogates what's going on through the human leukocyte antigen, the HLA complex, which is either class 1 if you're a CD8 positive T cell, or class 2 if you're a CD4 positive T cell. And those class 1 and class 2 HLA molecules actually display pieces of protein, pieces of antigens are put on the cell surface, and then the T cell receptor, with its full array of T cell receptors, can recognize what's going on. So these 2 pieces of biology, the left-hand side, the CAR, and the right-hand side, the T cell receptor, really represent where the field is at the moment in terms of engineering T cells to target solid tumors. So as we look at this therapy, one has to also put this into the context of 2 really important categories, because it's no longer sufficient to genetically manipulate cells. It has to now answer these 2 major questions. And the first is at the top, and that is the cost and the complexity of manufacture. How much does it cost, for instance, to make a CAR-modified T cell? How long will it take to make a CAR modified or a TCR modified T cell. And then the bottom, equally important, is to address what's going on either between patients or within the patient's tumor. And we'll hammer this home a little bit later, but this is known as inter and intra-tumor heterogeneity. And there are answers to this, and this is where Xyopharm, I think, has a real strength, and that is that one can address both the inter and the intra-tumor heterogeneity using what's called personalized TCRT or personalized genetically modified T cells, as well as the library approach where you're targeting essentially pan-cancer antigens. So companies, individuals, scientists that can address the cost and the complexity as well as the heterogeneity are going to have a winning formula. So what I've been working on for a long time, and Michelle, you kindly mentioned it back at MD Anderson when I started a lot of this work, as well as when I had the privilege of leading Xyopharm, was in the genetic modification of T cells using a non-viral solution known as the Sleeping Beauty system, okay? And this is a different way to genetically reprogram cells. And I've actually drawn this out on as you can see on the slide. So when one is using the Sleeping Beauty system, one is manufacturing DNA plasmids. For instance, that's the relationship that Xyopharm has with Aldevron, and then directly uses those DNA plasmids to genetically modify T cells to express, for instance, a CAR targeting, say, CD19, or a T cell, for instance, targeting an antigen that's represented in solid tumors. And that's in distinction from many, many others that are using viruses like lentiviruses, in which you still start from a DNA plasmid, but you have to take that DNA plasmid through a packaging cell line. You have to be able to produce conditioned supernatant that contains the virions that they then use then genetically remodify the T cells. So on the bottom of you can see the table of sort of the pros and cons of both sides, in that with the Sleeping Beauty system, it's cost effective because one's just simply using DNA plasmid without going through these intermediary steps before the genetic modification. It's rapid, it's customizable. You can cassette in and out your CAR, your TCR. And importantly, especially as Xyopharm is looking to its clinical trials in this space, one can essentially dial in all manner of T cell receptors targeting all manner of neoantigens. And that really will be important for those two pivots, if you would, the cost and the complexity, as well as addressing the heterogeneity. So the answer, not to give away the whole story for the presentation, is going to be using the Sleeping Beauty system and essentially having that rapid manufacture of clinical grade plasmid, such as with Aldevron. So why is the Sleeping Beauty system so useful in this regard? Well, it's not only its ability to cassette in and out the CAR and the TCR, but it has a property of being able to genetically modify resting T cells. So here's my attempt to show you that. So here is a Sleeping Beauty plasmid that's been electroporated. In other words, little tiny pores have opened up onto the cell surface, cell membrane of this genetically modified T cell, and then the DNA plasmid from the Sleeping Beauty system can get inside the nucleus. In other words, get across the nuclear laminin. That's not the case with a lentivirus or a retrovirus. When one has to transduce T cells to essentially stably integrate the contents, say a CAR or TCR, the T cell has to enter into a replicative cycle. In other words, it has to divide. And when it divides, the nuclear membrane dissolves, and that essentially allows the contents of the lentivirus or retrovirus to enter into the nucleus, and then you can get stable integration. That's a fundamental difference, and it's something that can be taken advantage of, as you'll see in a few minutes, in the Sleeping Beauty system, because you can electroporate resting T cells, and that's all you may have to do. And you don't have to put them in, if you would, in culture. So the Sleeping Beauty system I've been privileged to work on for a while now. We published the first clinical data in the transposon field when I was working at MD Anderson, and this technology was then licensed into Xyopharm for the targeting of CD19 specific CAR T, as well as other features. And there's been a lot of work since then. I just call out a few papers for the audience. But this work here in the CAR T space has actually gone on, and we've seen these patients now do very well who were initially involved, earlier, the company updated these data in 2020, to show that these patients are still doing well. And then, for instance, one of the lead scientists at Xyopharm, who did his postdoctoral work with Steve Rosenberg, Dr. Drew Denninger, he then adapted the Sleep and Beauty system to be able to express not CAR, but TCR. And that's essentially one of the drivers of why the company now is so successful in this field. So let's just spend just a moment examining where the company is in its CAR T world. And that is that it has made significant headway in addressing one of the major issues around CAR T, and that's the cost and complexity. That's, again, that kind of bubble, if we would, where we were on just a few slides ago. And the way it can be solved now is that one can use this non-viral gene transfer technology to simplify and scale out the manufacture. And you can see it schematized here. What the company's been able to do is take a leukapheresis product and express in the resting or quiescent T cells that are found in the peripheral blood, the CAR and other goodies that allow these T cells then to be rapidly infused, basically electroporated one on the first day, and then they meet release criteria and infused on the second day. The reason this technology works is that the transposon can get across the nuclear membrane. So we just kind of looked at that. But also, these T cells are given a special signal through what's called membrane-bound interleukin-15, or MBIL-15. And this allows T cells to be able to grow inside the human. And even small numbers of T cells can achieve, therefore, their therapeutic potential. So this is a step forward now in the commercialization of CAR T, because it essentially simplifies the whole logistical supply chain. You don't have to spend weeks in culture and all of the associated expense. And this is directly related to the DNA plasmids that are from the Sleep and Beauty system. And I would add here that the treatment actually is underway in Asia for this. And so this is clinical data that Xyopharm will be talking about in the months to come. So CAR T for CD19 is sort of the gateway, if you would, for T cell as a drug. But there's a real transition going on in the field. And you can see this in the lay press, you can see this in the academic literature, in that there's now a shift where CD19 malignancies are incredibly important, of course, and we must continue to hammer the cost and the complexity issues. But we also have to get to other cancers. So you can see that CD19 negative tumors are coming into the fore. These are hematologic malignancies. But importantly, the field is transitioning to solid tumors. And this is really the massive opportunity. And we kind of look at this on the left-hand slide, in that you can see, every year there's, 1.5 million or so patients in America who have invasive cancers. So to make this transition from CD19 malignancies down to solid tumors, I think there has to be a real switch. There has to be a switch then from CAR biology to TCR biology. In other words, we're expressing T cell receptors, not chimeric antigen receptors, to really capture this full opportunity. Just to kind of drive home why there's so much emphasis on going after solid tumors is that, again, there's just enormous numbers of patients. And you can see them in all types of cancers here, in the pie chart, and all the statistics on the right-hand side. So Xyopharm is motivated to essentially address this population, and it has the secret sauce. It has this DNA plasmid technology, the Sleep and Beauty system, already pressure tested in the clinic, to be able to now get into the expression of T cell receptors to get at the entire market. So let's now look at why this is going to be successful. So it's one thing to have the engineering principles, but it's another thing to be able to understand what you're shooting at. What are the targets that one's going after? And here we're really resting on one of the fundamental pillars of cancer, and that is the genetic instability of malignant cells. This is a hallmark of cancer. And that when a cancer cell divides and it has an error, as most cancer cells do, mutations that arise from that can be beneficial to that cancer cell. But they also represent an opportunity for the T cell to target those genetic mutations. So many or most mutations are unique to the cancer. That's important to remember. But a subset of these mutations are shared, and in other words, are in hotspots. So companies that are successful will be looking at opportunities for unique antigens, but also keeping in mind that they can target the shared antigens. And that's exactly what you're going to see in the next few minutes, is that Xyopharm has these two programs, a personalized T-cell receptor program to go after the unique antigens, and a library, if you would, program to go after the hotspot antigens. Okay. And just as some nomenclature here for the audience, and that is that when a mutation is able to tickle a T cell, in other words, a T cell can actually react to that mutation. That mutation is known as a neoantigen, a new antigen. Those mutations that are immunologically silent, therefore, are not neoantigens. They're just part of the background mutations that occur in that particular cancer cell. And of course, by now, you've seen through the presentation that the T cells are going to see these neoantigens, these immunogenic genetic changes, through their T cell receptors. And then essentially it's the ability of the T cell to then kill the cancer cell that allows the patient to go into remission. So I like to think of these neoantigens as the Achilles heel of cancer. Now, as part of the discovery of neoantigens, we know that these are tumor specific. In fact, by definition, they only occur in the tumor. That's important for the patient, of course, because when we're targeting neoantigens, we want to be able to reassure the patient that we're not going to have any off-target effects. In other words, the T cells are not going to trespass and inadvertently recognize normal structures. And as I've mentioned before, these mutations are sort of the seeds of their own destruction. That's where the Achilles heel nomenclature comes from. To address this, there's the two shots on goal, the personalized T cell receptor, as well as the library T cell receptor. And this targeting of neoantigens is based on a really rich literature now, in which we know, you can see with our own eyes, in fact, you can see it on your monitor right now, the patients that have had neoantigen-specific T cells can go into remission. And I put down the references here so you can take a look at the technology. Now, it's different from the genetically modified T cells. These are non-genetically modified T cells that are neoantigen-specific, and they provide an important proof of principle. That in other words, going after the neoantigens with infused T cells works, but it's not that scalable. And there are issues around the scalability because of the types of T cells that are being introduced. And we can kind of look at that here. So T cell therapy has established that kilograms of tumor can be treated in some patients using the infusion of targeting T cells. And particularly when the field had advanced from targeting germline antigens, which are not so effective, to when it's targeting neoantigens, these genetic mutations, then the field really stepped forward and said these are specific and these would have broad applicability. But the issue has been that in the non-genetically modified T cells, in other words, tumor-infiltrating lymphocytes that have grown up to large numbers, many of these TIL are ruined because they're unable to essentially recycle their killing, unable to get back into the tumor, to dialogue with the cancer, and to eliminate it. So therefore, we need a technology that essentially allows young, healthy, active killer T cells to be engineered to go after the solid tumors. And that's really where the Sleeping Beauty system essentially bridges the gap, because we know what's needed. We need to go after the neoantigens. We know how to do it through the T cell receptor. We now have to essentially be able to do it with killer T cells. And these T cells, thank goodness, are found in our peripheral blood. So now in your mind, you can see what you need to do. You take peripheral blood-derived T cells, you put in the right T cell receptor using this cost-effective and scalable solution through the transposon biology, And then that allows you to target either personalized unique antigens or shared antigens such as found in the library. And this essentially is where we're getting to now. And that brings up the other really important point of the presentation, and the one I really want to bring home, and that is that you have to be able to address heterogeneity. And heterogeneity is, of course, in two flavors, INTER, inter-tumor heterogeneity. And I made 5 little people here so you could see all the different shadings, if you would, or different types of antigens that are in their cancer. And so you need to be able to have T cell receptors that are faithful to each of the antigens within each of the individuals. And so you can now scale that by having different plasmids, for instance, different Sleeping Beauty plasmids that are expressing T cell receptors for each individual patient. And that's a match, right? That's a personalized immunotherapy. But of course, the other heterogeneity, INTRA, intra-tumor heterogeneity, has to be addressed. And here, for instance, you may have to put in multiple products, multiple T cell receptor products for each patient. And you can see I've tried to show that here, where you can see all the different colors that go up to be the collection of antigens represented by this particular patient's tumor. And now you would put in not one T cell receptor modified T cell, but 5 to match all of the different colors. Again, this plays right into the hand of a DNA biology because it allows you to scale for inter-tumor heterogeneity and scale for intra-tumor heterogeneity. And I'll bring this home again from a paper you can see published in 2013 in Nature that shows, again, inter-tumor heterogeneity, where you'd have the T cell receptors matching the neoantigens, or intra-tumor heterogeneity, where you'd have to essentially take account for all the little subsets of antigens that are expressed in this particular tumor mass, and that is infuse multiple T cell products, or for some special patients, to express T cell receptors that are against clonal antigens, such as KRAS or P53. So if we kind of look at that Sleeping Beauty system as the genetic tool, if we look at the heterogeneity as the challenge, now we can kind of step back and we can see why Xyopharm is really poised for success. Because on the left-hand side, we see the library T cell receptor process. On the right-hand side, we see the personalized T cell receptor process. So on the left-hand side, one's going after these shared neoantigens, particularly P53 and KRAS. And these patients then can be screened for expression of these antigens. These T cell receptors, because we know that these antigens are immunogenic, and we know that these antigens are drivers for cancer, these T cell receptors can be pre-identified and then use the Sleeping Beauty system to essentially express them in fresh, young, happy T cells from peripheral blood and infused to control the tumor growth. And this actually is now an active IND. It just got announced a few weeks ago for a clinical trial that's initially going to be run through MD Anderson. And then, of course, on the right-hand side, we have the more complex, but nevertheless really important personalized process that allows you to target a large number of patients with a whole battery of tumors. But in this situation, you need essentially to identify the antigens as well as the T cell receptors in real time. And this is where the Xyopharm team, the scientists, have really made enormous progress to simplify and expedite this process so that these T cell receptors can then be put into T cells, again, happy, healthy T cells from peripheral blood, and then infused into the patient. And this is also under IND, and this is the work that got started with Dr. Steven Rosenberg at the National Cancer Institute. So again, we have the two approaches. You have the library approach and you have the personalized approach. And I kind of took you through that on the last slide. And I'll just kind of call it out again. You're screening, you're selecting the right T cell receptor to match the antigen in the context of HLA. You're using the transposon system to stably integrate the T cell receptor, and then you're going through your quality control and infusing. On the personalized, a little bit more laborious in that you need additional steps to get the T cell receptor, but nevertheless, now this machinery is increasing in momentum to be able to, again, put in not just one T cell receptor, but put in a whole family of T cell receptors to look at the INTRA, the intratumor heterogeneity, and then again, meeting release criteria and infusing. So the process for the personalized approach is one in which it takes you through this code of conduct for identifying the antigen, identifying the autologous T cell receptor, and then infusing. And this gets at just a staggering number of patients each year may be able to be treated with this type of approach. And then we also have the library approach here. So the library approach, because it's in clinical, essentially it's under IND right now, I'll just spend a little bit more time on that. And just to call out, just in case there's some aficionados sharing this presentation here, that these mutations, these shared mutations in KRAS and TP53 are really important targets in that they are hotspots that are across a whole battery of tumors. And importantly, they're immunogenic. In other words, you can identify the T cell receptor to KRAS, you can identify the T cell receptor to TP53. And you can see some publications on your screen here that you can look at offline. The reason that KRAS and TP53 are being targeted by Xyopharm is that, again, they represent a large opportunity. These are some of the most important cancers. They afflict our brothers and sisters. And by being able to go after KRAS and TP53, large swaths of these patients can now have the potential to be treated with the Sleeping Beauty modified system, again, using these young killer T cells. So because the IND is now cleared, I can share with you some of the information about how this is accomplished. And the word that I'd like you to kind of take home is library. It's the first word in the title. In other words, these T cell receptors have been pre-identified. Just on the last slide, I showed you that rich literature explaining that KRAS and TP53 were well known to be oncogenic. So one can go about essentially identifying the T cell receptors ahead of time. You don't have to wait for the patient to show up. You know that woman with ovarian cancer is going to have a mutation in TP53. So let's get going. Let's start identifying the T cell receptor ahead of time. And that's just what Xyopharm has done. It has a library of about 30 T cell receptors targeting 18 different mutations in KRAS, TP53, and EGFR in the context of specific HLA combinations. Under the IND, 6 of those were curated and carried forward to be tested in the clinical trial. And then you can see that there's 3 each against TP53 and KRAS targeting the cancers listed on your screen. But of course, that library is going to be grown. And so that can be grown through in-house technologies. It can be grown through licensing approaches. And from the library, more of them essentially are curated to add to the 6. That makes strategic sense because the bigger the library, the more patients that can be treated. So this is a fundamental advance now for the company because it has this, if you would, sort of off-the-shelf library approach to go after these really important cancers. So here's some data that Drew Denninger and the team kindly provided me, where you can see several plots. Not to get too techie on everybody today, but what it shows you is that the genetically modified T cells climbing here on the y-axis. So now you have some genetically modified T cells. Almost looks a little bit like a weather map here. These T cells are in the upper left-hand side. In other words, they're not activated when they're seeing the wild type antigen, when they're seeing, if you would, the non-mutated antigen. But boy, when you give them a mutation, then these T cells are appropriately triggered. And you can see that as this shift occurs in the flow cytometry plot. You can see this as in the graphical representation here, as these red dots essentially show more and more of these activated T cells. And you can see this on the bottom in that these T cells with the appropriate controls are specifically lysing or killing only those targets that express the mutated antigen or the neoantigen. So kind of putting it all together now from the perspective of Zyopharm, there's really a complementary, unique set of technologies that belong to the company. And you just some words on the left-hand side to help you understand the graphics on the right. So the first is that they're using these peripheral blood T cells and they're engineering the T cells to express T cell receptors to redirect them to target tumors. And they're doing that through neoantigens. And they're using the Sleeping Beauty system, which is incredibly, incredibly important because it allows for essentially the swapping in and out of the T cell receptors to get at all of the patients with the T cell receptors that are needed in the personalized product. Again, multiple products per patient, and it allows you to get at the T cell receptors to build out the library. And this allows the company to treat just large numbers of patients with epithelial cancers that, again, just ruins the lives of hundreds of thousands of Americans every year. So I'll take a moment now, just before I leave you, and we go to the questions, just to kind of look at this from the supply and demand curve. Because again, it's one thing to participate in developing a technology like the Sleeping Beauty system that allows for the scalability, if you would, and the addressing the heterogeneity. But there's a fundamental shift that's going to occur, and this is really where Aldevron is really at the cutting edge, in that we're recognizing that the world is shifting. And this is a sort of an analogy here, but bear with me while I explain sort of how the cell phone, maybe I'll make a little bit of a pun here between cell phone and T-cell, but how the cell phone industry essentially somewhat mirrors where we are right now. So on the curve on the left-hand side, back in the day, if you would, when we were first getting cell phones, these cell phones were so expensive, so difficult to make, that only essentially a few products were available down here on the x-axis. And the expectation was that one was going to essentially have just a few products, but you're going to have just large amounts of sales because there just wasn't that choice, right? And in this situation, it really was difficult to commercialize the product because there just wasn't really the demand, if you would, curve. And what's happened in the cell phone industry, of course, is that Apple and others have come along to completely change this supply and demand curve. To where now, cell phones are relatively cheap, at least compared to, back in the 80s, right? And so there are many different types, if you would, of cell phones that are available. And there are essentially many products that are out there. So because the supply curve essentially has come down, because the cost of goods has come down, one can live, and this is the word that people use, in the tail of the long curve, where you can make many different types of products and have many people buy those products. And this tail of the long curve, in terms of quantity, just dwarfs where the industry was back in the 80s. Okay, a bit of a detour, but now let's just put this into reality today. So in the world of CAR T, as remarkable as it is today, it still costs way too much to make these therapies. And so we're living, if you would, back where cell phones were in the 80s. Those companies, such as Aldevron, such as Zyopharm, that are able to shift their manufacturing from just a few products to get large sales to where they're able to make a multitude of products, will be able to drive sales on this sort of individualized product by product basis and shift from the left-hand side here. I'll just sort of draw the cursor around it to the tail of the long curve. And this is where the opportunity lies. And this is why DNA biology is so remarkable, because it allows one to not only get at these incredible CAR T and these, the ability to have Lazarus moments, patients basically are saved from dying with B-cell malignancies, but it allows you to make the jump now to solid tumors with staggering numbers of patients and commercial opportunities, but to be able to do so at reduced cost and complexity so that one can get at the tail of the long curve. So, in summary, I think Sleeping Beauty provides the answer. It provides you the ability to scale, to express multiple T-cell receptors targeting multiple antigens. It allows you to address the inter and intra-tumor heterogeneity. There are trials at the NCI looking at this in the personalized space, and there are trials at the MD Anderson to look at this in the library space. So this is an exciting time for the field, and I just want to, on behalf of Xyopharm, I just want to thank Aldevron. They're just an amazing partner. The people is special, and the tech and their industry is just wonderful. So thank you much, and I'll yield back to Michelle now. We'll get to Q&A.
Great. Thank you so much, Dr. Cooper, for such an insightful and really intriguing talk about how you, through your previous leadership within Xyopharm and the team there, have really done a lot to think about not only the science and delivery to patients, but also really how to make this something that is attainable, affordable, and really in that time of need because of the just considering the impact and where these patients typically are in their journey. So thank you for that. This is the point now where I'd like to welcome and introduce my colleague, Dr. John Valentine, to the discussion portion of this. So a little bit of background about John. He's a co-founder of Aldevron since 1998, has been serving as our Chief Scientific Officer. And through his work, he's focused on collaborative research with our clients. He's also been responsible for much of the development of our processes and our assays. And currently he leads the research and development team here at the company. And they're seeking to develop new methods, products, enhancements on the services. And that still does, of course, include collaborations and technology licensing. So John, welcome to the discussion. Perhaps I'll turn it over to you to kick off the questions and conversation following Dr. Cooper's presentation.
Thanks, Michelle, and thanks, Laurence, for a great presentation. I think it encompassed a lot, obviously, on the scientific side, but also touched on some of the logistical elements that are going to be incredibly important to make this whole paradigm run. And I wonder if you could, if you were to say where we are now versus where you hope to be in the future, we're obviously at a place where manufacturing is going to be somewhat disseminated or distributed or, commoditized, delocalized, however you want to say it. Do you see any significant regulatory burdens with this type of approach, or is there precedent out there?
Yeah, I mean, that's a critical question. So, one of the things that Xyopharm did, when I had the opportunity to lead, was that it really dug not in on the clinical programs. In other words, standing up a CAR T program for CD19, but it used that to explore the regulatory science. And there's just an amazing regulatory team at Xyopharm that really partnered with me, and indeed partners still, to be able to understand, for instance, how one can rapidly release T cells. Because it's one thing to make genetically modified T cells, in 2 days, but it doesn't really help the patient if those T cells go through a week or more of inquiry in terms of how they're behaving. So I think one of the advantages is that rapid release testing, that science around that has been lockstep, if you would, with the genetic reprogramming of T cells. So that's a strength. But you also get at another important issue, John, in that as T cell therapy essentially gets to be shorter and shorter in terms of the manufacturing process, so there is an ability essentially to automate it, perhaps put into blood banks and so forth, and have a widely scalable model. And you can see, companies starting to look at that. And the reason, of course, is that it's just too damn expensive to have, a uber centralized manufacturing system that is shipping across the world. I mean, yes, it's today's technology, but it won't get us to where we need to go everywhere because it's very, again, the cost and the complexity of the supply chain. So the local manufacturer or the regional manufacturer is going to be really important. And there, for me, there's going to be two drivers for that. And again, it's kind of like back to the word heterogeneity. There's the heterogeneity of the patient. All three of us on this call look different. And it turns out that all three of us have different acting T cells. And when we put our T cells in a process, the donor matters, like what their T cells matter. And then there's also the heterogeneity of the process. In other words, what are you doing? What are you doing during your manufacturing steps to fiddle with the T cells, to engineer the T cells, and so forth? So those two heterogeneity drivers, the sort of complexity of the patient and then the complexity of the process, have to be clamped down. So to understand how to do that, you need just a kick-ass process development team. And Zypharm has that, Aldevron has that, and you can start to get in there and say, what's going on during my process and how can I tighten it and tighten it and tighten it? And then you need to figure out what are the patients doing? Who is the sort of the better class of patients? Or if we can't find a better class of patients, what can we do to make them a better class of patients? What other molecules need to be put in? What other genetic changes need to be put in to solve for the heterogeneity and get that tightened down? So as these two pieces come together, the process and the heterogeneity come together, and the manufacturing collapses down, then you have the magic moment. And the magic moment is when then it's push button. It's push button. You take simple plasmid, you put it into the T cells, and you can release those T cells according to defined criteria. And that's really where not just Americans will benefit, but we have to remind ourselves that 8 out of 10 kids with cancer are born outside of first world economies. And all of us, Aldevron and Zypharm, we have a responsibility for those patients. And so it can get to those patients, right? Then you can get to global impact.
Yeah, well said. Just as a point of clarification on the library approach versus the personalized approach, can you just expand a little on the likelihood or potential that the library could be the first step where it was followed by personalized, or are we talking about two divergent streams determined very early?
Yeah, at the moment it's two divergent streams determined early. It's because it's essentially just walking the road rather than running the road. But you're right, John. I mean, eventually, with the right type of screening technologies, if you would, a woman again with a p53 cancer could get her library T cells, right away, right? Because she would come in, she has a p53 driver mutation. The library is sufficient complexity to be able to match. But then you may say, well, does she need additional therapy? Does she need something down the road to perhaps cover for, an incomplete response or a relapse and so forth and so on. And that allows you to sort of get at the full spectrum, if you would, for these patients. But at the moment, it diverges. It diverges. If you got a library, you got to go on the library trial.
And also, just as another point, the little cartoon figure with the 5 requirement, we're talking about 5 different manufacturing events co-administered as opposed to a multiplex transfection.
That's right. Not 5 T cell receptors in one T cell. It's 5 T cells. I should have used my hand. 5 T cells, each with an individual T cell receptor.
So, Michelle, over to you.
Yeah, staying on the topic of the patient post administration, I thought I caught something there that the Sleeping Beauty approach combined with electroporation and really how that's approaching the cell drops the potential for the cytokine storm. Can you expand on that?
Yeah, it really comes from the CD19 literature rather than the TCR literature. So when large numbers of CAR modified T cells are put into patients with large amounts of circulating B cells, there's really sort of a clash of armies here. And that synchronous activation of the T cells then essentially produces all of the gamma interferon that drives, a cytokine storm that can be quite damaging, actually, to patients. The way around that is to essentially, borrow from the transplant literature. That's actually where I trained. And that is that small numbers of cells, in this case, hematopoietic stem cells, could engraft into patients and do so, and then essentially coordinate their anti-leukemia response, if the patient had leukemia undergoing bone marrow transplant. So the scientists at Zyopharm have understood that some T cells that are engineered with IL-15, remember that sort of membrane-bound IL-15 molecule, some of those T cells actually belong in a stem cell category. Again, there's some crossover between stem cell biology and T cell biology. So people call these sort of stem cell memory T cells. And there is somewhat, of sort of a promised land, because they have an ability to sort of be evergreen. They can grow and nurture themselves in the stem cell state, but also kick off progeny that are killers. So the concept here is that you can just need just a few numbers of T cells. And those T cells then will grow in the patient to match essentially the biology. And you don't put two warring armies together. You put, if you would, little, sort of ninja T cells of small numbers, but can do remarkable amounts of benefit.
You're really good at the analogies, Dr. Cooper. So just one last question, I'll pop it over to John, but you have me thinking about, a patient population in a very dire, challenging situation, informed consent, getting that right. How is that piece going? I mean, the educational aspect, this is high science, these are big concepts. I'm just curious of the response that you're getting from patients and their caregivers.
Yeah, no, it's, I think that's one of the really rewarding parts of, when I left academia to join Zyopharm, is that, MD Anderson, nation's #1 cancer center, is a remarkable organization. I love it. But you were treating patients within my orbit, right? I was treating patients in Houston. People were coming, but it was in the transplant world. And I saw a bigger vision for the transposon technology. I saw a bigger vision for what we might be able to do to sort of personalize T-cell receptor therapy, get into solid tumors. But you need large amounts of capital to do it. So that's the real transition to industry. But that essentially, that launching pad now has been very gratifying. We were able to launch a company out in Asia, a joint venture named Eden Biocell, with James Wong, who's now the lead director at Zyopharm. And so there's a whole transposon world going on over in Asia. There's Zyopharm now that has, important clinical trials going on at the NCI and at Anderson. And there'll be more to come in terms of being able to get this out there. So from the patient perspective, it's certainly going in the right direction. But of course, proof's in the pudding, right? Like all things, the CAR T worked really well. We understand that, patients are, benefited from the infused T cells. And I fully expect the same thing to happen with the T cell receptor T cells. But that's why we need the clinical data. And that's why Zyopharm, we'll get that clinical data, get these patients on the trial, and we can see how well all of this technology works.
That's great. A long history of engaging together. So it's absolutely rewarding, to your point. John, I will pass it back over to you before I have one last question for the two of you.
Yeah, I guess one of the things that conceptually is out there, and of course the numbers become very large, is the potential to front run a library of 1 million mutants. Mm-hmm. Obviously the cost associated with that is probably refractive to what most people would consider to be a reasonable deal. But can you speak to that as to whether that would be a library or a repository of significant enough amounts of DNA to make this work? Is that a viable approach if there's enough money printed to do it?
Yeah, and that's really interesting. So at the moment, where Zyopharm's focused and a lot of the industry is focused is on pan-cancer antigens, EGFR, KRAS, P53, and a few others, right? And that makes sense because the upfront and strategic investment, for instance, with Aldevron, is one in which there's a defined universe of T-cell receptors against a very large number of HLA molecules. So it's still a big work product if we were to get the totality of those patients. But one can turn the next card over, and the next card over are where these shared antigens are tumor specific. For instance, DIPG is a very important histone mutation where it only occurs, we think, in DIPG, small numbers of patients, thank God, but one could essentially map out the entire mutanome of those mutations and then generate all the T cell receptors against all the HLA. That's a known piece of biology. It can be industrialized. I think where the meeting of the minds will occur is that scientists like yourself at Aldevron will develop technologies to make small numbers of DNA molecules available for each of these individual patients, and then those DNA molecules could be on the shelf for the entire catalog, if you would, of mutations that are either shared between cancers or living, for instance, in a particular cancer. And that's a day that will come. For sure it will come, because once it's shown that P53 is a target, KRAS is a target, the natural next step will be to say, well, what else is shared? What else can we put on the library? It may not be so common, but for those patients, it'll be really important. And if we can get the costs down, again, that supply-demand curve, then the tail of the long curve just keeps on going. You know what I mean? And you get more and more patients on, maybe in the onesies and twosies, but incredibly important for them, but drivers of economics for industry.
Yeah, it's almost a stratification, if you will. The more that we learn, we'll know what what we consider maybe frequent flyer, items, and then kind of from there, what's next, and then could get some, from a manufacturing perspective, some level of kind of restocking that shelf, if you will, based on prevalence, et cetera.
Yeah. And I think that's right. I think the other sort of small wrinkle there is there are going to be patients for which they're truly going to be unique.
Yeah.
They're going to, but there again, the relationship between Devron and the sort of T cell companies can be fundamentally altered for the better through time, because this is one of the things that already benefits actually Zyopharm, is that when Zyopharm calls up and said, oh, I need a T cell receptor, the machines of Aldevron can turn fast enough to get those individualized products made, right? And so it's not just the library gets made ahead of time for all of these patients, but it's also for the individual patients that will never benefit from the library because they truly are unique. Then essentially you can make those DNA plasmids super fast.
And the library benefits them.
Yeah. Totally benefits them. Yeah. Mm-hmm. Sorry, Michelle, I think I cut you off.
No, that's great. I mean, actually, your mention of DIPG, it perfectly tees up kind of my last question for the two of you. I had a neighbor who was impacted by DIPG, and I know that there's just really nothing out there for those kiddos, and it's devastating for families. And so my last question to the two of you is just more of a blue sky, knowing the potential of this technology, of what we're starting to now see clinically. What are you most excited about, as far as just the future and the outlook for patients?
Yeah, I'll go first. I mean, just so we'll use DIPG as an example. So Michelle, I'm sorry for your neighbor. So actually, Zyopharm does have a clinical trial in DIPG using IL-12 biology. This is something that is I think very important because there's just nothing out there. But it's an example. So one of the future statements is that the genetic engineering of T cells to express a CAR or TCR is just the first step down the road, because as essentially libraries of T cell receptors become available, as the manufacturing costs get under control, the heterogeneity gets solved for, you can put more things into a T cell. And you can start putting cytokines into a T cell. And you just saw an example, with IL-15 being in a T cell. So, in terms of that rapid 2-day manufacturing of CAR T. So one of the things that I'm optimistic about, for Zyopharm in the field, is that we'll start seeing molecules that redirect specificity married to other molecules like cytokines to get a product that can solve for additional complexities in the tumor microenvironment. Because after all, hallmark of cancer is genetic instability. These tumors are going to be, devil themselves, right? They will try and sidestep the immune response, even though it's going to be aggressive and directed. the tumor will fight back. So in this arms race that we're in terms of T cell therapy, it's having the redirected T cells as well as ancillary molecules that will be able to solve for essentially that pushback that the tumor will inevitably have.
Yeah, I think my blue sky thing, Michelle, is we talked a bit earlier about the ability to distribute or democratize manufacturing out to the coalface. And, you know, we always like to think of having that Coke machine sitting in the corner that you plug a sequence into and come back the next day and there's a vial with what you need in it. A lot of the technologies that exist to make that happen are sort of macro right now. It doesn't mean that in the future we couldn't have let's say a synthetic step that's point of origin is the plasmid that's just been isolated in a very small quantity from the coli as a jump off point in a very small flow path in a completely disposable system, and it would allow that cancer center to be making that treatment right on the spot. It would shorten, it would diminish chains of custody, handoffs, complexities associated with multi-site manufacturing. And I think that would be the ultimate incarnation of where we need to get to in this space. It's going to progress in small steps, but without those small steps, we'll never arrive at what is the ultimate in deployability, and that is for a big cancer center or a small cancer center near a cell shop to be able to make the things on demand, literally. And I mean, that would be the perfect scenario. Obviously, we'd like to be making the enzymes for that. But then the other part of it is towards the library. Every time a construct is made for anyone's personalized indication, you make an extra couple of milligrams, and you expand the library that way. Over time, and I can see that being a potentially a way of chipping away at this to build up more and more, and with a singular site where there's availability, maybe across multiple CMOs, where it's sitting in a freezer, it's all the paperwork's good, and a hit shows up in Florida or Washington State, and that evening that DNA is on its way from wherever the point of storage is to that site, and triggers a resupply that's on a time scale that can fit in for any manufacturer into their vacant slots. That would be a great way of restocking, and it would as long as enough people in our game were willing to get involved in this, we could quite quickly build up a big supply of a large number of epitopes. So.
I'd actually up the ante on your blue sky, John. I think you're absolutely right. The rapid availability, the almost plug and play, if you will, once you know sequence, having that available on a global scale, kind of to Dr. Cooper's previous point, when we can reach third world countries and have an impact there, that I think is a tremendous blue sky moment. And just hearing the presentation from Dr. Cooper, the discussion subsequent to that, I think the pieces are definitely there. This is such an exciting time. I'm grateful to both of you for sharing in really making sure that we can bring this forward to our audience that's going to be listening in. So thank you both for sharing your time. Thank you for your presentation, Dr. Cooper. And then for the audience, I really encourage you to look for future Aldebaran Breakthrough Speaker Series events that we'll be hosting. So thank you again. Have a great day.
Have a great day.
Thanks, Laurence. Thanks, Michelle.
Thanks.